Hello friends and welcome to my channel Sequential Biology. In previous video we have discussed about fertilization process in human and sea urchin, and in this video we will discuss what is cleavage and characteristic features of cleavage. And after fertilization the zygote of the many animal species undergo a succession of the rapid mitotic cell division called the cleavage or the cellulation or the segmentation. So the zygote undergoes mitotic cell division. So cleavage is nothing but this is a type of mitotic cell division by which single cellular zygote will be transformed into multicellular morula and the blastula. So this special type of mitotic cell division is known as cleavage. We will see few important points of cleavage. During cleavage, the cell cycle primarily of the M phase and the S phase. That means only the division and the DNA synthesis can be seen during cleavage, and the no G1 and the G2 phase can be seen. Cell shows two type of phase, the interphase and the mitotic phase. The mitotic phase is a normal division, and interphase is the preparatory phase of mitotic cell division. And in interphase there are present three subphases, the G1, G2 and the S phase. In G1 and the G2 phase the growth of cell can be seen, and in S phase the DNA synthesis is occurred because mitosis is a type of equational cell division. But during cleavage the cell cycle can be seen, but a special kind of cell cycle can be seen during cleavage. Only S phase and the M phase can be seen during cleavage, though no G1 and the G2 phase can be seen. Since no growth is not occurred in resulting cells, so the cells become smaller and smaller during the time of cleavage. The result of the cleavage is formation of the morula. Then the size of the morula will be equal to the size of zygote, but this morula contains multicellular structure, but the volume of the zygote and the morula remains same because the cells of the morula are resultant of mitotic cell division of cleavage. But since during this time the no growth phase can be seen, so the cell will becomes smaller and smaller, right? And the cells which are formed by the process of cleavage are known as blastomere. Then during cleavage only in cell cycle you can find the two phases, the S phase and M phase. Then G1 and the G2 phase are skipped, and the little or no protein synthesis can be occurred during cleavage, and there is no increase in mass, and the cleavage partition the large cytoplasm of the zygote into the many smaller cells are called as the blastomeres. So by the process of cleavage the many smaller cells are formed, and these cells are usually known as blastomere. The first 5 to 7 cleavage division produce a hollow ball of cell which is known as blastula. Then the first 5 to 7 cleavage division produce a hollow ball of cell which is called as the blastula, and surrounding a fluid filled cavity which is known as blastocell. Then this is a zygote, you can say this is zygote, and by repetitive mitotic cell division this produce a multicellular structure which are known as morula, and morula is a compact multicellular ball like structure. The cells remains attached with each other by a special kind of protein which is known as E-cadherin. So E-cadherin are present just like this, and this will hold all of these cells, right? And later this morula will be transformed into the blastula, and inside this blastula a special type of cavity can be found, and which is known as blastocell, and the surrounding layer of this blastula are called as blastoderm. The cells of this blastoderm then undergo morphogenetic movement to produce the gastrula, which contains different type of germ layers. Anyway, the result of the zygote is formation of multicellular unilayered blastula, right? At onset of the cleavage it is quite rhythmic, but later it become irregular or asynchronous. So this is another important characteristic features of the cleavage, and the resultant cells are smaller. By the process of cleavage many blastomere are formed, but these resultant cells are usually smaller. The total volume of the cell mass remains unaltered with the volume of the zygote, that is no growth can be found. So the resulting structure or the multicellular structure, the volume of the multicellular structure remains same with the zygote, because during the cleavage no G1 and the G2 phase can be seen, so no growth can be occurred during cleavage. And the cleavage cell cycle is very faster. Since the G1 and the G2 phase are skipped, so the cell cycle is very faster during the process of cleavage. In next video we will discuss the rules of cleavage, and here we will only see the characteristic features of the cleavage. So what is cleavage and what are the different characters of the cleavage, right? Now the characteristic of the cleavage. The cleavage, the zygote undergoes the mitotic cell division, then all the division are mitotic and that occur rapidly one after another, and the cleavage is more than propagation propagating multicellularity also determine the cell fate. That means cleavage determine that what type of cell will becomes in what structure, right? And this is a very fast division since the node G1 and the G2 phase can be found in during cleavage. So these division are very fast, so fast that the S phase of the next cleavage begins already in the telophase of the preceding one, right? And you know the M phase is consisting of the 4 phase, the prophase, metaphase, anaphase and the telophase. But before completing the telophase, the resulting cells again undergo the synthetic division or the S phase, right? So the cleavage rate is very fast. The early cleavage division occurs synchronously and the division one after other, right? So this is called synchronous division. This is very important. Early cleavage division occurs synchronously, the division one after other. All the blastomere divide simultaneously and the late cleavage are asynchronous. When the time proceeds, the late cleavage remains the asynchronous, and the cleavage increases number of cells but decrease the size of cell. This is very important point. Cleavage increases number of cell but decrease the size of cell. And during the cleavage, the reduction in the nucleocytoplasmic ratio, that means the resulting blastomere in each successive stage, the nucleocytoplasmic ratio will be decreased. The cell become more active because the nuclei have less cytoplasm to control. This is very important. And here you can find the volume of nucleus and the volume of cytoplasm. At 4 cell stage, the volume of nucleus is 1, but the volume of cytoplasm is 18. And when the cleavage process proceed, then at 64 stage, the volume of cytoplasm will be reduced, but the nucleus remains the same. Then the high amount, so cell become more active because nuclei have less cytoplasm to control. And here in 4 cell stage, the nuclei have greater cytoplasm, 18. So 18 is greater than 12, right? So nuclei have greater cytoplasm to control in 4 cell stage. But in 64 cell stage, the nucleus have less cytoplasm to control. So this 64 cells becomes more active. And in blastula stage, the ratio of the nucleus and the cytoplasm will be 1/6. So this become again, the nucleus have very less amount of cytoplasm, so this blastula remains very active, and the cells of blastula or the blastomere undergoes morphogenetic movement, and this will causes the formation of gastrula, which contains 3 germ layer, ectoderm, mesoderm and the endoderm. And the no growth of the cell, so no quantitative change in the cytoplasm of the blastomere can be seen, and the blastomere do not move, and here the blastomere do not move, so the general shape of the embryo does not change except the formation of a cavity, the blastocele in the interior. So in morula, the morula actually absorb the fluid, and this form a internal cavity which is known as blastocele. So during this time or during the time of cleavage, only the blastocele are developed in blastula. Chemical conversion of the reserve food material into the active cytoplasm, so yolk and the glycogen will be converted into active cytoplasm, and the cleavage convert egg into a compact mass of blastomere called the morula. Then the cleavage convert egg into a compact mass of blastomere called morula, which ultimately transform into blastula, having unilayered thick mass of blastoderm around central space called the blastocele, right? So this is the result of cleavage. Now if we see an example of the sheep, what will be the result of the cleavage? The result of the cleavage is formation of morula and the blastula, and here you can see how morula and the blastula looks like. So the zygote undergoes mitotic cell division, and first it becomes 2 cell stage, and later it becomes 4 cell stage, and ultimately this will produce a compact ball of cell, and which is 16 cell stage, and this 16 cell stage is called as morula. There is no cavity inside this morula, and later the cells of the morula absorb the fluid, and this will form a structure and which contains a cavity inside this ball, and this is known as blastula, and this cavity are called as blastocelic cavity. And the blastula have a covering, and it is known as trophoectoderm, or this is called as epithelial cell sheet, and this trophoectoderm later will form the extraembryonic structure. And with this trophoectoderm, the few cells are hanging, and these cells are known as inner cell mass, and ultimately from this inner cell mass the embryo will be formed. And the cells of trophoectoderm which remains attached with this inner cell mass are called as cells of Rauber. Okay, so ultimate result of cleavage is the formation of blastula. Then how blastula are formed by the sheep, we will discuss it, and this is very important, and you should have the concept, you should have this concept that how the cleavage leads to first differentiation event. The zygote is a totipotent stem cell. Totipotent means it has the capability to generate all types of cells in our body. Stem cells are usually three types: the totipotent, multipotent, and the pluripotent. So totipotent cells are those type of cells which can form or which can be differentiated into all types of cell in our body. So this type of stem cell are called as totipotent stem cell. And multipotent stem cells are those type of stem cell which can be differentiated into many type of cells in our body, but not all. Like hematopoietic stem cells will generate different type of blood cells in our body, but not all types of cell in our body. So hematopoietic stem cell is a type of multipotent stem cell, and unipotent stem cell which will be differentiated into another type of cell. Like RBC progenitor cell will be transformed into only RBC, not another type of cell. So this is called as unipotent stem cell. And the stem cells also have two important property, that is division and the differentiation. But the zygote is a totipotent cell because it has the capability to generate all cell types in our body, and also the inner cell mass, also the totipotent stem cell, any cells of the inner cell mass are responsible for formation of all types of bodies. Then the zygote and the inner cell mass are usually the totipotent stem cell. Now the fertilization quickly followed by cleavage. In mammals, the cleavage occur before embryo implanted. This is very important. And you know, embryo are implanted in our uterus. Actually, if we draw our female reproductive system, you can find the fallopian tube. So this is the fallopian tube, and fertilization occurs in the ampulla of our fallopian tube. So fertilization causes the formation of zygote, and ultimately the zygote try to implant it into the uterus. So this is the uterine wall, and anchoring of this zygote into the uterine wall is called as implantation. But just after fertilization, the cleavage process will soon begin, and this will produce a multicellular structure, and before implantation a multicellular structure is formed, and this is known as blastula, and ultimately this blastula will be implanted into our uterine wall, right? Then the fertilization quickly followed by cleavage, and in mammals the cleavage occur before embryo implanted. In sheep, each cell at 8 cell stage has potential to give rise to a complete animal. That means this 8 cell stage are totipotent stem cell. The 8 cell embryo divide again. What will be the nature of this division? The mitotic cell division or the cleavage. Okay, so this will form the 16 cell stage morula. The embryo undergoes compaction due to the surface molecule or the E-cadherin. So more compact the cell structure morula is formed, right? And the next, some cells adhesion are diminished and the fluid begins to flow into an internal cavity, that is the blastocell are formed. An additional division of the cell produce the 16 cell stage blastula. And here you should notice that how many type of cell can be found in morula and how many type of cell can be found in blastula. Then the zygote first form the 8 cell stage, right? So this is 8 cell stage, and the morula is 16 cell stage. So these are important. Then the morula contains 16 cell stage, and these cells are remains compacted by a special kind of protein which is known as E-cadherin. And the blastula contains 16 cell stage, but like morula, but in this blastula a fluid filled cavity can be formed, and which is known as blastocell. Then the blastula are separated into two cell type, the trophoectoderm and the inner cell mass. Then the differentiation or the separation can be seen in blastula, right? But in case of morula, no differentiation can be seen. Then the trophoectoderm, which will form the extraembryonic tissue like the placenta, and the inner cell mass which give rise to embryo proper, and the source of the embryonic stem cell which can contribute to any part of the embryo, and the inner cell mass expresses a special kind of gene which is known as OCT4 gene. Both inner cell mass and the trophoectoderm have the property of the stem cell, and each type of cell divide to renew itself, right? So inner cell mass is located on the side of blastocell, then inner cell mass are located on one side of blastocell, and inner cell mass are loose mass that can be described as the mesenchyme. Inner cell mass are also known as mesenchyme because these cells are very loose mass of cell. And the trophoectoderm form a hollow ball around the inner cell mass and the blastocell, and they organize to form the epithelial sheet, and they will be implanted. So this structure will be implanted. Then the zygote which is formed after fertilization is not implanted because the zygote is a unicellular structure, and this will undergoes many repetitive mitotic cell division, and ultimately this will form the blastula, and this blastula will be implanted in our uterine wall, right? Then what will be the target of cleavage? The target of cleavage is formation of blastula, and this blastula will be implanted in our uterine wall, right? Then these are actually the characteristic features and few basic idea about cleavage. And in next video we will discuss rules of cleavage, and sometimes you becomes confused when you read about rules of cleavage, and in next video we will discuss about the rules of cleavage, and after then we will discuss the types of cleavage, and later at last we will discuss the cleavage process in frogs and chick, right? So this will be actually the sequential series of cleavage, and hope you had understand about very basic concept and characteristic features of cleavage. So thank you, and that's it.